In Chapter 14, we're going to take a look at the evolution and human health. What is it that evolutionary biology can teach us about human health and disease? So they start off by looking at some examples. In this case, we're looking at cholera. And we're going to look at the idea that we can use evolutionary biology to help us understand these. And so what you're seeing here is an evolutionary tree of this cholera epidemic. And we can tell from this particular tree where the cholera that infected Haiti, where it came from. We know that it was introduced to Haiti from Nepal because we can see that when we isolate the strains of cholera from these people, that these strains are more closely related, i.e. more similar DNA, to the Nepal group than they are to any other group on the tree. This is one of the ways that we can trace the origin of a particular strain of virus and then its path of movement. So if you wonder how they were able to track, let's say, where coronavirus entered the United States and how it moved through there, that's one of the ways that we can do it, is we compare these samples, we look for similarity in DNA, just like we would if it was an evolutionary time scale, because in many ways, for viruses, this is more of an evolutionary time scale for them. And so we can trace that. So we can definitely use evolutionary biology to understand the origin of particular strains and the order in which they diverged geographically. So this is a valuable tool that we have in medicine. Think about this pathogen, the virus or the bacteria, as a population. And in that population, we will see different genetic makeup, different alleles. And remember that evolution occurs within that population. One of the things we have to think about when we think about human health, particularly human health from a pathogen perspective, is there's two different populations. We have the population of the pathogen, so the cholera or the influenza A or the coronavirus. We have the pathogen, but then we also have a population of cells within our own bodies. We actually have within patient population, and then we also have this pathogen population. These two populations are clearly going to be in an evolutionary conflict, because what is best for the pathogen is clearly not as what is best for the patient. The pathogen is under selection to reproduce very rapidly, and in doing so, by nature, because it is a pathogen, it's going to take resources from the host to be able to make those. And then, of course, the host is under selection to limit that, to limit the damage by slowing or killing that pathogen. So we definitely see this evolutionary conflict. So what is it that makes pathogens so successful? One of the things is that they have very large population sizes. So if you look at a typical colony of bacteria or a viral population size is huge. They also have very short generation times. And so they can produce more and more individuals in a very short period of time. And going along with that, they also have very high mutation rates. So those things together means that in a very short period of time, you can accumulate a lot of different mutations. And what that means is they evolve very quickly. So we looked at these things before in Chapter 1 when we were looking at HIV. And they, of course, apply here as well. One of the things we find, though, is that the hosts are actually going to impose selection on the pathogen, because the host will launch defense mechanisms against the pathogen, and that pathogen over a very long evolutionary history has been dealing with these defense mechanisms. We can think of our defense mechanisms as a selective agent. Pathogens evolve around these defense mechanisms because these defense mechanisms are selecting organisms that are able to do so. One of the things that we find then is if we impose very, very strong selection, only the best of those pathogens survive. So very, very strong selection, but we also have very high variability in our pathogens. So even though we have very strong selection, there will be some out there that are capable of overcoming our defenses because they have that variation. That means that evolution will occur very, very rapidly. If you remember from the chapter when we looked at response to selection, remember, response to selection was looking at the amount of variability, genetic variability you have in a population times the strength of that selection. And here we have both. We have lots of variability and very, very strong selection. So this evolution will occur very, very quickly. So let's take a look at how and why these flu populations evolve. Fitch study investigated whether the human system, immune system, is actually responsible for the change that we see in the influenza A virus. So we see the virus changing. Is that the result of dealing with our immune system, or is it evolving for other reasons? If we know that it's evolving because of our immune system, this might help us develop better and more effective vaccines, because we can understand what those selective pressures are and kind of use countermeasures. If they aren't evolving in response to our immune system, it's some other factor, then we need to take that into account too. The influenza A is responsible for annual epidemics, so this is the flu shot that we see every year, is this vaccination against these new influenza A viruses that evolve. Occasionally, these will become global pandemics. These flu viruses can be. Ordinary flu season kills approximately 20,000 Americans. If we compare that to the 1918 flu pandemic, anywhere from 50 to 100 million people over a several-month period were killed by that pandemic. This is what the influenza A virus looks like. To understand some of these different strains that we're going to look at and the evolution of it, we have to know a little bit of its biology. What we see is two major surface proteins. So here we have sort of this coat on the outside, and we have two major surface proteins, the hemagglutinin and the neuraminidase. Hemagglutinin initiates infection by binding to the host cell. This will be like a recognition type of a protein, and it will allow it to recognize particular types of cells. However, it also then acts as sort of a marker for our immune cells to recognize it. So it allows the cells to bind, but this is also a flag, if you will, for our immune system to identify this foreign entity. The neuraminidase is another one of these surface proteins that we have. In addition, we have inside of the virus 8 pieces of RNA that make up the viral genome. So this is the viral genome. It would make some of the key proteins that this virus needs. And of course, what's going to happen once this virus gets into a cell, it will use the cell's host machinery to make more of these viral RNAs and then to reassemble this complete virus, very much like we saw with HIV. So we can see that these different 8 pieces of RNA actually encode for 13 different proteins. This is the hemagglutinin, and it has 5 antigenic sites that are showing in color. And the antigenic sites are the sites that our immune system would recognize and remember. So if it has these particular sites, our immune system recognizes that site and then can launch an attack against that site and can form memory to that site. So how does an influenza A virus stay alive in light of the fact that our immune system will attack it? In essence, what it has to do for this pathogen to stay alive is it has to constantly change these antigenic sites to try to essentially trick our immune system to not be able to recognize it. So if we can swap out some amino acids in some of these antigenic sites and change them just a little bit, then we can essentially confuse the immune system, at least temporarily, where it cannot recognize those antigenic sites. Fitch et al. Hypothesized that flu strains with novel antigenic sites are actually favored by selection. So that makes sense. If I come back here and I change this antigenic site, now the immune system does not recognize it, and now this particular virus can continue to replicate. They used viruses that were isolated from humans in this time period from 1968 to 1987. From an evolutionary perspective, from the virus's perspective, that's a very long time. Viruses evolve 1 million times faster than we do. So in the virus's world, this is more of a time span than we have between when humans and chimps split from one another. So that's a long time span in the evolutionary history of a virus. What Fitch did was he went in and he sequenced these hemagglutinin genes and then estimated the rate of evolution, then created a phylogenetic tree from those rates. We see the amount of divergence from the original sample over time. Basically, every time we change up this line, we're acquiring new mutation. This mutation is occurring at a pretty constant rate and quite rapid. That's a pretty steep slope, but it's fairly constant rate. So over time, we see a pretty constant, rapid rate of evolution. Down here, what they did is looked at some of the different branches from the influenza A viruses, and note that most of these lineages represent extinct side branches. So a whole bunch of these different lineages came off the side, but they don't continue down. When this stops and it doesn't go all the way over, that's an extinct branch. The strains that did persist were all very closely related to one another. And what they did then is they looked at, so of the ones that persisted, what was similar about them? What he found was that they had replacement mutations within those antigenic sites. Where we find those mutations, it's not just a random set of mutations that makes it. The set of mutations that makes it are ones that are altering the antigenic sites. And of course, these are the ones that allow them to evade the host defenses. Follow-up studies find this from other flu strains as well, where most of the strains that make it have mutations in these antigenic regions. Can we use this information to then develop vaccines? So what they do is they try to predict where the change is going to occur. So we know, for instance, there have been 18 codons under very positive selection in the past. So we can predict that the most successful viruses in the future will have mutations in that region. And so what they've done is they've gone in and then predicted what will, say, next year or the next few flu seasons, where do we think those mutations will be? And this method has actually allowed them to accurately predict 9 of the 11 recent flu seasons as far as where do we think the changes will be and what kind of vaccine would be effective for that. So they are able to make some of those predictions and get more effective development of vaccines. So in the next video, we'll take a look at what happens when these flu viruses like influenza A reach global pandemics. What patterns do we see in these global pandemics?